Hello student, welcome to EPG Pathsala. I am Dr. Ajit Jaiswal from Department of Anthropology, Pondicherry Central University, Puducherry. Today we are going to discuss about a module called DNA Technology and Medicine under paper Physical or Biological Anthropology. The main learning objective of this particular modules are: in this module we are going to learn about the DNA technology. We will try to know about the various types of DNA techniques and the application of DNA technology in medicine. Let me give you an introductory information about this module. In view of the emerging complex diseases that are hindering the quality of life of human population, anthropologists, in particular biological anthropologists in the recent past, focused to synthesize the concept of biological anthropology in the study of human disease that vary from population to population in order to promote the health of the human population. DNA technology have revolutionized the application of molecular genetics in understanding the population specific gene expression, susceptibility to a particular disease, evolution of abnormal gene, understanding the gene expression and its variation from population to population, allowing devising gene specific drug therapies. Human being can't escape from the disease as it is a part and parcel of life. Even though a very tiny portion of populations are inherited with certain mutant allele, but a majority of the human develop mutant allele due to exposure to the environment. Exposure to the environment over a period of time enables man to develop certain gene abnormalities that are identified as a degenerative disease. In view of the rapid growth of industrialization and urbanizations, innumerable changes are taking place which are witnessed in the form disease prevalence in mid or early 40s. Addressing this issue became a great challenge for the scientists including biologists, biological anthropologists. Even though the quality of human life is dependent on the genetic endowment, but the interference of environmental factors like social, cultural and biological factors on gene expression override towards abnormalities. Demarcations of health hazard due to gene environmental interaction need to be tackled on priority basis. In view of the diversity in cultures and lifestyle across population, it is imperative to understand the DNA and its changes in a specific culture in order to promote and implementing the effective interventional strategies towards the welfare of human. What is DNA technology? DNA technology is a revolution in biology and is having ever increasing impact on clinical medicine. Earlier genetic disease could be tracked through pedigree analysis and affected protein. However, no confirmative pathways could be achieved through this technique. Thus, a new technology which can directly study the DNA is warranted. Deoxyribonucleic acid, what we call as DNA, otherwise the organism genetic material, is inherited from one generation to another generation, holds many clues that have unlocked some of the mystics behind the evolution. Disease participation, aging and exposure to the environment. Manipulation of genetic material is called as DNA technology. DNA technique makes it possible to take any gene from any species and place this gene in any other organism. DNA technology has plethora applications in our day-to-day life. DNA technology will be very much useful in identifying certain genetic disease such as sickle cell anemia and Huntington's disease. Thus, there is an ample possibility that certain genetic disorders or diseases can be tracked and treated before its precipitation. DNA technology facilitated to develop a vaccine which triggered the defense mechanism in the body to fight against the pathogens. Further, the development of therapeutic hormones like insulin and human growth hormones are the resultant of DNA technology. Cloning. Cloning, a major component of DNA technology is cloning, which is the process of making multiple identical copies of a gene by incorporating in an organism like bacteria to use in a different application. This identical or multiple copies of DNA can be characterized or used for various purposes. Cloning gives us pest resistance plant, vaccine, heart attack treatment, and even entirely new organism. Polymerase chain reaction, it is also called as PCR. Polymerase chain reaction represent one of the most significant discoveries or invention in DNA technology, and it led to a 1993 Nobel Prize award for American-born Carey Mullis. PCR is the amplification of a specific sequence of DNA so that it can be analyzed by scientists. Amplification is important, particularly when it is necessary to analyze a small sequence of DNA in quantities that are large enough to perform other molecular analysis such as DNA sequencing. Now let's discuss about the recombinant DNA technology, what we also call as genetic engineering. Not long after PCR technology was developed, genetic engineering of DNA through recombinant DNA technology came into limelight in parallel with the PCR technology. Altering the DNA by using bacterial-derived enzymes like restriction endonuclease that act like scissors in cutting the specific site of DNA is called recombinant DNA technology. This DNA sequence can be inserted into another matched DNA sequence. Restriction endonuclease play a very important role to identify the variability between the two individual or genes, etc. Restriction enzymes that recognize specific DNA sequences can produce fragment of DNA by cutting different parts of a long strand of DNA. If there are differences in this sequence due to inherited variations, restriction enzymes no longer recognize the site. A variable pattern can be produced. Gene therapy. Recombinant DNA technology also helpful in measuring the genes into molecular devices that can transport this gene to various cellular destinations. This technique also called as gene therapy. In this technique, corrected genes can be delivered into individuals with defective genes that cause specific disease. Different bacteria can be modified through these techniques aimed at breaking the harmful contaminant like DDT by providing specific protein. Currently, efforts are in progress to produce genetically designed plant and crop that can produce substances to kill insects. Plant food can be engineered to have genes that produce specific protein which slow the ripening process by increasing their shelf life. Many heart and autoimmune diseases have been treated by using this gene therapy. This technique hopefully tackles the issue of Huntington disease or cystic fibrosis by replacing the defective genes with healthy ones. When we discuss about DNA microarray technology, this technique, also known as DNA chip, a latest development in nanotechnology, is to study the genome in high throughput manner. This technique facilitates gene expression profiling to understand the specific gene up and down regulation. The up and down regulation of gene allow us to estimate the cancer risk because it has the ability to know the gene expression that is above the baseline level. However, it cannot detect the delicate changes in gene expression that causes specific disease. When we talk about DNA profiling, methods that are used to describe DNA profiling include restriction fragment length polymerase RFLP and short tandem repeat profiling, that is STR. In RFLP, DNA will be cut into different lengths by restriction enzyme, and then the segments are separated based on size through electrophoresis. STR will facilitate many copies of a small section of DNA by using a specific enzyme. Genes from one organism can be inserted into another organism to produce nutritional and pharmaceutical products. For example, cow genes have been modified so that it can produce vitamins or insulin in its milk in bulk. Similarly, plants gene also modified to produce desired quantity, taste with added nutritional value. This will play a major role in future in increasing the supply of both food and energy needed by the world's growing human populations. Application of DNA technology. DNA technology has revolutionized biology and is having an ever-increasing impact in pharmacology, genetic engineering in disease prevention, in increasing agricultural growth, in detection of disease and crime, etc. Some field that has shown remarkable growth due to advance in DNA technology include forensic, bioinformatics, pharmacology, nanotechnology, archaeology, and anthropometry. DNA extracted from archaeological species can be used to address anthropological questions. These help in tracking DNA evolution, migration pattern, and species evolution over the ages. Through the DNA technology is relatively new, even though the DNA technology is a relatively new area of research, but it has enormous debate. It will likely continue to be public debate and have its impact on medical diagnosis, therapeutics, forensic, and genetic profiling. DNA technology in medicine. The emergence of the DNA technology has been driven great strides in understanding fundamental life process and the ability to investigate problems that had previously been unapproachable. The advances made possibly by DNA technology have profound implications for the future of medicine, for they have placed us at the threshold of a new method of diagnosis, prevention, and treatment of numerous human diseases. Hormones, vaccines, therapeutic agents, and diagnostic tools developed using different DNA technologies are already greatly enhanced medical practices. DNA technology has aimed a commonplace in day-to-day life. A new product from genetically altered plant, animal, and microbes became available for human use. Example: Dolly, a sheep in 1997, stood in headlines as the first successful cloned large mammal. Thereafter, many advances have been taken place in medicine towards treatment. Example: cancer. Now, because of cloning, many organs and tissues can clone. Scientists vision that DNA technological applications are one of the new frontiers in science with tremendous growth and disorder potential. DNA technology made it possible to treat different diseases by inserting new genes in place of damaged gene in the body. Thus, it has brought many changes in the field of medicine and introduced such method of treatment and disease drug delivery. The first two commonly prepared products from recombinant DNA technology are insulin and human growth hormone, both of which are cultured in the E. coli. Patients with diabetics fail to produce requisite amount of insulin, facilitating the processes of sugar from food into energy that the body can use. In the past, patients used to take insulin obtained from pigs and cows. However, non-human insulin often causes allergic reaction in many diabetics. With the advent of recombinant technology, now we could able to produce safe human insulin. In this technique, human insulin gene is isolated and inserted into plasmids, which are transformed to either bacterial or yeast cell, wherein insulin is synthesized on the human code. The purified insulin is identical to human insulins or absolutely safe. Human growth hormone is a polypeptide hormone. It functions for proper growth, reproduction of cells and regeneration. It is secreted by somatotrope cell present in the pituitary gland. In recent years, DNA technology has helped to develop many growth hormones. Dwarfism can be effectively tagged with this technique. Monoclonal antibodies are used to produce vaccine against different viral infection. When body is encountered by foreign object, immune system of the body release specific protein called as antibody. Hybridoma technique made it possible to produce monoclonal antibodies where lymphocytes or B cell joined with myeloma cell to produce hybridoma. The antibodies released by hybridoma is called monoclonal antibodies, which are used to produce vaccine against several viral infections. Antibiotics are the substances used to fight against bacterial infection and other microbes causes infection in the body. Alexander Fleming discovered penicillin for the first time in 1928 using the recombinant DNA technology. Similar to insulin technology, edible vaccines to prevent widespread disease in developing countries could be achieved effectively through DNA technology. Edible vaccines are cost-effective, easy to manage and store, safe and socio-culturally readily acceptable vaccine delivery system. In this technique, desired genes are inserted into plant and then inducing this altered plant to manufacture the encoded protein, where it is vaccines or other plant substances. Initially, it is thought to be useful for preventing infectious diseases only. However, its application has reopened in prevention of autoimmune diseases, birth control, cancer therapy, etc. Successful expression of foreign gene in plant cells and or its edible portions has given ways in developing plant expressing more than one antigenic protein. Multi-component vaccine can be obtained by crossing two plant lines harboring different antigens. Adjuvants may also be co-expressed along with the antigen in the same plant. B subunit of Vibrio cholerae toxins (VCB) tend to associate with copies of itself, forming a donut-shaped 5-member ring with the hole in. This feature can bring several different antigens to microfold cell at one time. For example, a trivalent edible vaccine against cholera, ETEC, enterotoxigenic E. coli, and rotavirus could successfully elicit significant immune response to all three. Global Alliance for Vaccine and Immunization deals very high priority to such combination of vaccines for developing countries. The other medicinal product as a result of DNA technology are as follows. Later, several such products to elevate different conditions were come into existence. For example, tumor necrosis factor in the treatment of certain cancer. Certainly there is a immune deficiency and HIV infectious treatment. Then porokinase in the treatment of heart attack, taxol in the treatment of ovarian cancer, and interferon in the treatment of viral infection and certain cancer. Further, DNA technology allowed development of many tests which are being used to diagnose diseases like tuberculosis and cancer. Similarly, other diseases like measles, smallpox, hepatitis, this can be diagnosed by using this application. In the diagnosis process, certain pathogens are isolated and identified, and then diagnostic kits are produced. When the genome of the specific pathogens is known to eradicate or block its pathogenic activities. Now let's discuss the advantage of this DNA technology in human being. DNA technology has the ability to trace the disease history. Besides management, every individual in one way or the other are subjected to a condition or disease. This may be the result of inheritance of a certain disease from parent or alteration of phenotypes due to environmental mutagens. These mutations causes several diseases like cystic fibrosis, several heart disease, chronic infections. These abnormalities can be reverted by exercising the DNA technology by replacing the bad gene with a functional copy of the gene in the correct position. This technology will be helpful to slow down their aging process. By understanding the genetics with the help of genetic engineering, we can develop better pharmaceutical product for human subsistence. Disadvantages of DNA technology. DNA technology has a certain disadvantages also. Basically, genetic engineering uses viral vectors to carry functional genes into the body, and the consequences of the viral genes on human body is yet to be elucidated. The location of the functional gene in genome is also not known. Otherwise, this functional gene may replace other important genes rather than the mutated gene, which may lead to new complication. The replacement of defective gene with functional gene may lose genetic diversity. Hence, this technology has to be carefully applied, keeping the advantage and disadvantage. The advantage like DNA technology has the ability to trace the disease history. Beside management, every individual in one day or the other are subjected to a condition or diseases. This may be the result of inheritance of certain gene from parents or alteration of phenotypes due to environmental mutagens. These mutations cause several diseases like cystic fibrosis, heart disease, and chronic infections. These abnormalities can be reverted by exercising the DNA technology by replacing the bad gene with functional copy of the gene in the correct position. This technology will be helpful to slow down the aging process. By understanding the genetics with the help of genetic engineering, we can develop better pharmaceutical product for human subsistence. Now let's try to give you some important highlighting features that we learned in these modules. First, DNA technology has revolutionized the application of molecular genetics in understanding the population specific gene expression, susceptibility to a particular disease, evolution of abnormal gene. Next, even though the quality of human life is dependent on genetic endowment, but the inference of environmental factors like social, cultural, geographical and biological factor on gene expression override towards abnormalities. Next important point, in view of the diversity in culture and lifestyle across population, it is imperative to understand the DNA and its changes in specific culture in order to promote and implementing the effective intervention strategies towards the welfare of human being. Even we also try to learn about DNA, deoxyribonucleic acid, otherwise the organic genetic material is inherited from one generation to another generations. It holds many clues that have unlocked some of the mysteries behind the evolution, disease precipitation, aging and exposure to the environment. Even manipulation of genetic material is called as DNA technology. The DNA technology has a plethora application in our day-to-day life. Different DNA-based technology are like cloning, polymerase chain reaction PCR, recombinant DNA technology, DNA fingerprinting, gene therapy, DNA microarray technology, and even DNA profiling. DNA technology facilitated to develop vaccine which trigger the defense mechanism in the body to fight against the pathogen. Further, the development of therapeutic hormones like insulin and human growth hormones are the result of DNA technology. The emergence of DNA technology has been driven great study in understanding fundamental life processes and the ability to investigate problems that had previously been unapproachable. The advance made possible by DNA technology have profound implications for the future of medicine, for they have placed at the threshold of new method of diagnosis, prevention and treatment of numerous human diseases. Thank you.